Now let's look at those macroscopic view of translation, the protein synthesis. Protein information. The amino acid sequence is matching to RNA sequence, right? Then there should be some connection between RNA sequence to protein sequences. Now here, the machinery of making the protein is so-called ribosome. This is ribosome, and this is mRNA. And this is protein synthesized. Between protein synthesis to mRNA, there is another connecting motif here that is so-called tRNA. And this mRNA template is moving through from one side to the other side and passes through these machinery systems. Then tRNA is carrying this new amino acid is coming through here and then connect that to the next chain of growing polypeptide here. Now the connection of these two happening through this matching. So this is genetic code, and 3 units of this genetic information coding one amino acid. That's what it said. To connect this exactly matching to this, now this complex has tRNA has complementary sequence to this genetic code, and then they can match here, and AAA match to UUU, G to C, A to U, U to A. So that's why genetic code is GAU. tRNA should have complementary code CUA, and that's how this is connected to each other. By looking at this picture, can you figure out which one is 5', which one is 3', and which one is the N terminal to which is the C terminal? You can see here, this is incoming amino acid, and this is a product. Can you guess which is the starting point and which is the ending point? Yes, this is the starting point, and then the amino acid is growing to these directions. So then we can call maybe this is N terminal, and this is a C terminal, and then this is a 5 terminal, and this is a 3 terminal, 3' terminal. Then we can say this mRNA is moving from this direction to this direction, moving out, and then protein is synthesized. That's why this sequence is matching to this sequence here. Clear? So this is 5' end, this is 3' end, and this should be N terminal, and this should be C terminal. This is the structure of ribosome. Ribosome may be the biggest machinery system in our body, and the size is bigger than 20 nanometer. And we will see those comparison of different the size of different machinery later. And it has 2 subunits, one large subunit and then one small subunit. And this is a top view. Let's check the several factors about the ribosome. As I said, ribosome consists of 2 units, one large unit and one small unit. And ribosome is complex with a ribosomal RNA and dozens of different proteins. Ribosome structure is quite conserved among all different species, and small prokaryotic ribosome is about 20 nanometer size, and eukaryotic is about 30 nanometer size. RNA contents is about 65% for prokaryotic ribosome, and for eukaryotic, the protein portion is increased, so it's almost 1-to-1, half RNA and half proteins. Important thing here is the contents of ribosome. The main component is not protein, it's RNA. If you think about the function of ribosome, ribosome is the catalytic machinery to make a protein. In a sense, that's essential or most important function in life forms. By reading genetic information to make out the old proteins, old machinery system is done by ribosome, and it is not protein. The main component is RNA. So that's why this is the basis of RNA world hypothesis. And RNA can be enzyme, so that's why this can be called as a ribozyme. And maybe in the ancient time, the earlier the life form may have only RNA without the DNA or the protein, and RNA can serve both as genetic information and at the same time catalytic the enzymes. Prokaryotic the ribosome is composed of 2 units, so total complex is about 70S, and each unit is 50S and 30S. Small unit has 16S RNA, which has 1,500 nucleotide and 21 proteins. Large subunit actually has 2 components of RNAs, one bigger one 23S and 5S, and bigger one is almost 3,000 nucleotide and about 30 proteins. Now here, something looks very strange, isn't it? So let's say here it's a 70S and it's combined with the 2 components and 50 and 30. And if you add 50 and 30, it should be 80, not 70. Something doesn't match. So here the S is not the standard unit. This S is sedimentation coefficient. So that means under high spinning the conditions, like in centrifuge conditions, and this defines the speed of sedimentation sitting down. So high speed means it can sitting down faster than lower numbers. So this is approximately connected to the size, but it's not linearly correlated to the size. But this is experimentally defined, and this is a commonly used unit for this kind of huge other macromolecules. Maybe 20 nanometer size may not give you a practical idea. Then let's compare different materials size. Average size of a human being is a little taller than 1 meter. And if you go down here, most of those proteins is smaller than 10 nanometer. Lipid molecules, you can think those thickness here, and atoms is a sub the nanometer size, so 0.1 nanometer is 1 angstrom. Carbon to carbon bond is approximately 1.5 of the angstrom. Then you can think 1 nanometer is about 10 chemical bond is connected together, and most of those proteins are smaller than 10 nanometer. Now we said 20 nanometer or 30 nanometer of ribosome size. This is much bigger than common protein size. This is even close to maybe some small virus. Virus is approximately 100 nanometer or smaller. Now 20-30 nanometer is comparable to virus size. Just a single machinery system in the body is a similar size to virus. It's quite big, isn't it? Common cells in our body, those are in the range of 10 micrometer to 20 micrometer size here. And some subcellular organelles such as mitochondria or maybe the prokaryotic the life forms like bacteria, these are in 1 micrometer the range. So this is the good to know. Cells, mammalian cells, eukaryotic cells, about 10 micrometer size. Bacteria and mitochondria about 1 micrometer. And virus is about 100 nanometer. Proteins are smaller than 10 nanometer. And ribosome is a 20-30 nanometer size here. So let's look at a little more details of ribosomal or the small unit. Here the blue color is protein and orange color is RNA. Easily we can see the central scaffold is made of RNA and the proteins are accessory motif attached surrounding those RNA scaffold. So we can assume that these proteins are doing some regulatory job, but general structure is from RNA. How about the larger unit of ribosome? Larger unit has 2 RNA, so one is designated as orange color, one is yellow on top, and still they are forming the central structure. And you can see those blue structures are proteins attached to the RNA structures. Utilizing this, the ribosome, now let's look at those dynamics. Here, this is a small subunit of ribosome. This is a larger unit. And then mRNA is passing through here, moving this direction to this direction, go in. Same direction, those tRNA should come here too. They are carrying this amino acid, and then they are attached here. And that amino acid is transferred to the growing proteins. So here, which is the N terminal, which is the C terminal? This should be synthesized first, and then this is moving to this direction. That's why this is the N terminal, this is the C terminal. How about here? Which side is 5' and which side is 3'? It should be this side is 5', this side is the 3' at the end. Now, mRNA is moving out to this direction, and then this tRNA is coming in, and then this amino acid is transferred to this guy. Now we can think of two different ways. This peptide was attached here on P site, and then this guy is moving out. Either this amino acid is transferred from here to this, or this peptide is transferred from P site to A site. Which one is happening here, do you think? Chemically speaking, maybe it's easier to transfer small unit to the larger unit. But actually in biological system, interestingly, this long chain is transferred from P site combining to A site amino acid here, attack here. And then this guy is moving, and the next one is coming in, and the whole peptide is moving from this side to the next one. That's how it works in protein synthesis. Now let's look at a little more dynamic movies here. So here, the ribosomal subunit is combined together, and then you can see here RNA is moving from one direction to the other, and then new tRNAs are carrying in amino acids, and then they are attaching here. If you look at carefully, then one move here, and then that whole structure is transferred to the new other tRNA. And then synthesized the protein is growing out, and through this hole it is pushing out to the outside. Okay, here I add one professional of the movie for RNA synthesis and protein synthesis. Please look at very carefully, especially for those amino acid coupling to growing polypeptide.
When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, how was it? Did you see how their amino acids are coupled to each other? Okay, here is the quiz 1 for you. So please check the question and give your answer on Paul in Ivy League.